And good or bad, to humanize us a little bit, Dan's a Eagles fan, so I don't know. I knew that guy would like that, that's for sure. So,
And unapologetic about it.
okay, perfect. Let's get you your slides here. All yours.
All right, thank you. Thanks for coming out so early and finding the room. I know I had a little difficulty finding the room this morning. Today, Bruce and I are going to talk to you about a grand challenge confronting members of the agricultural community, scientists, agribusiness, farmers, employees of the USDA. And that grand challenge is feeding 9 to 10 billion people by the year 2050. And the role of geoinformatics, more particularly the geoinformatics platform being developed by ARS, our agency, the Agricultural Research Service, in meeting this challenge. The informatics platform being called AgCross. Now, to feed 9 or 10 billion people, we have to dramatically increase food production. We have to increase the nutritional quality of food. And we also have to decrease the impact of food production systems on the environment. More food, higher nutritional content, while at the same time maintaining the quality of the water, the soil, the air. We need to produce this food. Now, to meet this challenge, scientists have to change how they do science. We've got to break down silos. We've got to collaborate. We have to change the way we use existing technologies, and we have to develop new technologies, more environmentally benign technologies for food production. Playing a major role in all of this is geoinformatics. Now, let's see if I can, there we go. Now, it's been estimated that we have to increase food production 60 to 100%. Again, we have to produce more nutritionally dense food. Past food cultivar, I'm going to talk mostly about plants. Cultivar development is focused mostly on increasing yield, macronutrient content, fat, carbohydrate, protein. We also have to enhance the nutritional quality by increasing micronutrients in the food. Provitamin A in the case of plants, B complex, vitamin C, E, K, amino acids, and fatty acids the human body can't produce. We need to get it from nutritional sources in our diet, as well as, 23 to 29 different minerals. We get all this from our food, and plants are a primary provider of all this, either to us directly or to animals that we eat. And oh yeah, we have to do this very quickly. 2050 is not that far away in terms of science. We make a lot of mistakes on the way to getting things right in terms of technology development. And we've got to do this while at the same time we're confronting climate change. Altered weather patterns, drought, flooding, changed temperature extremes, too hot, too cold for crops. Hot temperatures dramatically decrease the photosynthetic efficiency of major crops all the way around the world, used all over the world. Much above 30 degrees centigrade, photosynthetic efficiency drops off dramatically. Elevated temperatures, high levels of CO2 also have a very negative impact on nutritional quality of food. We've got to develop crops that can deal with these conditions. We can't simply double the acreage for agriculture. A lot of competition with urbanization for arable land. There's going to be even more because we're going to have more people. We're losing arable land to poor farming practices. Soil fertility is going down in a lot of cases. And you can't simply repurpose landscapes to agriculture without having impacts on the environment, particularly biodiversity. The Green Revolution everybody's heard of, started in the 60s, doubled food production, solved a lot of world hunger, intensely used synthetic fertilizers and pesticides, high-yielding crop varieties without any focus on micronutrient content. However, fertilizer feedstocks for a lot of major components for plants, macronutrients are dwindling. In some cases, a lot of environmental degradation associated with the Green Revolution practices, soil erosion, decreased soil fertility, eutrophications of our lake, pollution of groundwater, rivers, oceans, dead zones. Got to change this. We need a second green revolution. Part of this will be using the existing technologies that we have, synthetic fertilizers, synthetic pesticides differently, more efficiently. Geoinformatics plays large in this in precision ag. What geoinformatics allows you to do is stack layers of data, georeference that data. In the case of this slide, soil type, soil nutritional qualities, NPK, last year's yield. You build a model, you inform the farm equipment as it goes over the field. At precise locations, it delivers the right amount of fertilizer or pesticide in that case. You're decreasing resource inputs, you're maximizing yield, you're maximizing economic return, you're minimizing impact on the environment. We need to develop more environmentally benign crop production tools, biologicals, cover crops. Stephen's going to talk about this extensively. Biologically based disease controls, beneficial microbes, fungi that colonize other fungi and kill them, bacteria that kill plant pathogens. And then we have to use the same tools to efficiently utilize these more environmentally benign tools, precision ag, and Stephen will talk about that. For example, here's a next generation cropping system for tomatoes developed in our group, the Sustainable Ag Systems Lab back in Beltsville, Maryland, where hairy vetch, a cover crop, was switched in to replace black plastic, the industrial standard. For this production system, you make raised beds, you plant hairy vetch in the fall, it overwinters as a cover crop, you mow it and kill it in the fall, and then you transplant directly into the mulch. Here you see a comparison of the two systems, black plastic, the two leftmost images. You see a lot of exposed soil, you get erosion. With the hairy vetch all the way to the right, you get a lot of ground coverage, actually complete ground coverage. You decrease erosion. Other benefits associated with the hairy vetch is that it's a legume. It fixes nitrogen. You cut down on your nitrogen needs for tomato. Tomato has a very high nitrogen demand, so you're cutting down on the synthetic fertilizer you need. It suppresses disease. On the east coast of the United States, early blight caused by Alternaria solani is a big problem. It's a soil-borne problem. Soil-borne plant pathogen. When you have rainfall events, you get splash dissemination of the inoculum from bare soil into the tomato canopy. Causes disease. When you have hairy vetch completely covering the ground, you impede the splash dissemination, you reduce disease. Reduces erosion and runoff, you already saw that slide. Reduces pesticide runoff because you have better water infiltration. Pesticide collects on this black plastic, gets washed off. So you're cutting down the pesticide load in the runoff. Suppresses weeds, kind of not the best. It's economically feasible, which is extremely important. No grower is going to use it unless they get good return on their dollar. And oh yeah, it impacts tomato physiology. You get delayed senescence. Tomatoes yield far into the fall compared to the black plastic system. And you also get induction, for some reason, we haven't figured it out yet, of plant defense genes that result in the synthesis of osmotin and chitinase, two antifungal compounds produced by a tomato. Beneficial microbes, more environmentally friendly than synthetic pesticides. Here we see on the right sclerotia of a very important plant pathogen, Sclerotinia sclerotiorum, a soil-borne plant pathogen, again, completely colonized by this mycoparasite, a fungus that grows on another fungus. It kills the sclerotium. That sclerotium produces the initial inoculum. By killing that sclerotium, you reduce the initial inoculum, you get less disease. So if you spray the field with this mycoparasite before you sow the crop, you cut down on disease by cutting down on initial inoculum. And we use this in place of synthetic pesticides or along with reduced rates of synthetic pesticides. Another beneficial microbe we're working with is Serratia. It's got these really cool red colonies. What we do is we extract the biomass or the cell mass of Serratia with ethanol. And that works as well as a seed treatment with a synthetic pesticide or when you spray the field in controlling disease caused by pitheaceous fungi. So we have other tools we're developing. If we combine this with precision agriculture, we can use them efficiently. And again, of course, precision agriculture is very dependent on geoinformatics. Geoinformatics are important in developing advanced crop germplasm. Allows you to look at the genetic landscape. So breeders and molecular biologists can produce advanced crop germplasm that yields when confronted with these temperature extremes or biotic pests such as plant pathogens. Also, you can make crops more nutritious. And then finally, geoinformatics helps scientists to change the way they do science. They can be more collaborative. ARS has a number of different, what we call high PI driven projects, or at least what I call high number of PI driven projects, one of which is LTAR, Long-Term Agroecosystem Research. It's conducted at 18 different sites throughout the United States, representing a number of different agroecosystems, croplands, rangelands, pasture lands. What scientists on this project are tasked with is developing new management technologies based on ground-based sensors, remote sensing, computer modeling, and development of mobile technologies, decision support tools. Geoinformatics, of course, plays a major role here. Another large project conducted at a number of different sites throughout the United States is GraceNet. The objective here is to decrease greenhouse gas evolution from crop production fields, sequester carbon. In all, there are over 60 different experiments conducted at over 35 locations based on geoinformatics being conducted by ARA scientists. And what this geoinformatics platform, AgCross, does is connect these scientists. Oh, bad color choice there. Streamer bad. Anyhow, AgCross allows collaborative efforts, free exchange of scientific data. Becomes a repository, it should become a repository of all this geoinformatic data. Will allow all this participating scientists to get the most advanced analytics, push them out to the scientists, and also become a test bed for development by computer scientists of next generation analytics. What it'll allow us to do is change the way we do science. In the past, going back to the precision ag example, you have all these different layers of georeference data. You make your model. The model could be very good for this precise location. You publish a paper, and then where does the data go? It's stuck with the scientist. with AgCross and the repository for all this geoinformatics, the data is stored. Scientists can use data created by other scientists. You can grab the data, makes your model more powerful. You have advanced analytics, the model's more powerful. You have more transportability of your model to other systems. Now, Bruce is going to talk to us about the vision, ARS vision for AgCross, this geoinformatics platform.
Thanks, Dan. I'm going to show you the architecture. I'm the IT architect on this system for AgCross. In the center, you see AgCross. AgCross is built upon ArcGIS Hub with a backend tied to ArcGIS Online. We have an Azure presence that has a backend SQL POS with enterprise geodatabase on top of it. It also ties to the LTAR website. And then as we look towards the outer part of it, there's an interconnection with Ag Data Commons for DOIs and metadata, which in essence is pushed up to data.gov. As we look at the new developments on the left-hand side of the screen, we have the ARS imagery server, which is Esri's imagery server. We have an ARS sensor server, which is a GeoVid server. And then we have the FarmBeat servers, which these guys will tell us what's in those at some point in time here. There's a few Microsoft guys there. And then at the bottom, we have on-demand artificial intelligence, AI, and analytical server tools. They can both be transported into AgCross or use AgCross data or also be pushed to the stakeholders. And as you see on the very outside, we're utilizing farm crowdsourcing, imagery, sensor inputs, and the Internet of Things, FarmBeat UAV. Up at the very top, we have Internet 2, SciNet, and the series high performance computing with an AI. With that, we're going to go back and forth via Globus to the imagery server, or we'll be able to push up into it from AgCross. Up at the top, we're utilizing Esri operation dashboards, insights, and others. We're bringing data in. We're going to be bringing data in via Esri Survey 123 and Collector. So I'm going to give a live demo now. Okay, so what we're going to look at here is AgCross. It's broken down now by network. You see the antibiotic resistance, the Dairy Grand Challenge, which is DAP. You see the GraceNet, which Dan alluded to. You see LTAR. You see Nuonet, and you see REAP, which is Resilient Economic Agricultural Practices, and then the Soil Health Network. One of our things we're going to do to try to break down some of these silos is we're going to be exploring by topic going forward. So we could look at cover crop, we could look at soil biology, we could look at greenhouse gas. So let's go into AgCross overview. And what we have here is we have all the sites that are within AgCross right now, across the country. If we go into one of those sites, it will allow us to bring up a picture of the people and allow us to go to that particular site. This is what's being measured, this is what's being shown on the site. So if we open up the table of contents, you can see all the layers that are within that site and what they do. If we go over to these tabs here, you can see insights. And do this, make it a little bit bigger, a little bit smaller. So what we have here is we have it distributed by treatments. Each of these are a treatment here. And outside are the units, the plots which they are on. So if you click on one of these, what it will do is it will filter all the experimental unit versus the CO2 that's being used. It will show which units they are, and it will show graphs accordingly on that. You can do that with N2O, you can do that with CH4, etc. We're going back to AgCross again. We can also look at greenhouse gases. So let's take a look at greenhouse gases. Let's look at the CO2. So here what we have is we have all the experimental units, average across all the experimental units of mean CO2 gases across all the sites within across to measure greenhouse gases. So I'm been told that I'm going to be cut off here shortly, so let's go quickly to LTAR here. And Stephen's talk is going to be part of what's going to be shown in this one here. Oops. They are at the Lower Chesapeake Bay LTAR site. And if you go to the cover crop pilot project, what we have is what they're doing here on the cover crop project, and he'll be showing more on this as we go through. You hit the layers over here. You'll be able to see what's in there, turn on and off layers. He'll be showing that as well. And I think at that, for right now, not to cut time, I'm going to say that's it for right now, and we'll move on to Stephen.
Any questions for our speakers? I have a question. So this is great research, right? This is good stuff. Is there going to be a point where there will be either a sharing of the results of this, or are you looking for a collaboration with private industry as well?
Which research you're referring to?
Your choice. I mean,
We do both.
okay.
We publish. We work for the U.S. government, so it does belong to the people of the United States, and we're working with Esri right now and Microsoft, as a matter of fact.
And AgCross is built on open data, so all the data within there is shared.
Okay, so people get access to it.
Get access to it, download it, do whatever they want to do with it.
Fantastic.
And we're basically going to build it out to be able to do models on it and be able to do analytics on it down the road.
Great. So I guess
We're
what
1 year in, so.
Yeah. Oh, yeah, understood. What I'll do is we'll talk about this, get the link to that, and I'll share it like in that LinkedIn group that we talked about. That way, those people can see it, or if you guys join that group, then you can get access to it. We'll go from there. All right. Thank you very much.